Okay, everyone, it's time for us to go ahead and start Lecture 10 for this week, or Lecture 10 for the class. And so it won't be too much longer. Can you believe the classes, we're getting to the point where we're almost done? Time flies way too quickly. Anyway, I hope your projects are going well. I'm hoping y'all got some good feedback on your projects, okay? And so remember, we're adding to a web page. You can do a PowerPoint if you wish. You can do a paper if you wish. You could actually put together a web page, but I'm not expecting you to. That's something I plan to do with your information. If you have any questions or you need anything, let me know. I do plan on popping into office hours tomorrow if you need to chat with me. I do have a bunch of appointments with students, so if I'm not there right away, I will be there. And so if I'm not there, please send me an email and I'll make sure to check in for you guys too. Okay, so hopefully everything's going okay. Just relax. We're on the downhill slide. And from what I can tell, y'all seem to be doing great. So we are actually going to cover the topic today called de-extinction. And I'm not talking Jurassic Park, by the way, which gives me a headache every time I see the movie. I think I've told you all this before, that I used to work in the field of ancient DNA. And so there are so many reasons Jurassic Park will never happen. First of all, amber is a horrible preservative for DNA because it's very porous. So if we do manage to get dinosaur DNA, it will not be out of insects that are saved in amber. I had friends who actually tried to publish this in Real Science, and all they were ever able to amplify is bacterial contaminants. Okay, so I do know that not too long ago, a wonderfully preserved sample of dinosaur was found, and they actually found some proteins, which was a big deal, that were preserved, since 65 million years ago. But again, science has a long way to go. And, if we do manage to resurrect extinct species, it's not going to be through amber, okay? Can you tell my husband has no fun going to movies with me? Because I start critiquing everything and then he just gets annoyed. But anyway, sorry, it's a scientist in me. So let's talk about de-extinction in real life. Okay, so back in the 1970s, there was a species of frog known as the gastric brooding frog. And the reason it has this name and why it's actually so interesting is the fact that mom frogs, when they get pregnant, basically lay their eggs and then they consume them to protect them. Okay, but ultimately what happens is so the mom frogs will convert their stomach into a nursery. This means that all of the gastric acid and so forth has to disappear. And so the mom frog, hopefully before she lays her eggs and consumes them, will ultimately eat enough food beforehand that can last her, because once her stomach converts into a nursery, there's no more getting any nutrients, okay? But back in the 1970s, researchers had the foresight to realize this frog was endangered, and so they took a sample and stuck it in the freezer. Now, the freezer is the best way to actually preserve DNA. What scientists have been able to do with this frog is they've been able to use what's called cloning, and the process of cloning, to create an embryo. Now, I don't know that anything has gone further than that, and I don't think the embryo ultimately hatched. However, they're basically getting really, really close to bringing back an entirely extinct species. Now, this can happen in more than one way, but let's go ahead and talk about the process of cloning and why this can be used for such an endeavor. Now, you might have heard of the process of cloning with regards to Dolly the sheep, because she was one of the most famous and largest critters of her day to be cloned. So let's talk about how this happened. So if you think of sexual reproduction, we're talking about the boring version, okay? You have an egg and a sperm come together that each have half of the genes of their parents. And so when they come together, they create a fully complete chromosomal creature, okay? Now, what scientists did is they didn't necessarily reinvent the wheel, but they found a really slick way to use this in their favor to create what's called a clone. So what they did in sheep was they took an unfertilized egg cell. You can see that at the bottom left. They pulled out the nucleus, which normally only has half the number of chromosomes as it needs. So then they have this empty egg. Okay? Now, move to the very top of left, and you see that there's a critter that is going to donate its genes or its genetic pattern. So they pull out a cell from that individual, and they take out the complete number of chromosomes, and they insert them into this empty egg. Okay, so this egg no longer has half the number of chromosomes, it has a complete set of chromosomes. Then they add some chemicals and biddity-boppity-boop. Sorry, I was watching something with that today. But the critter starts to then, the zygote okay, which has a complete set of chromosomes, starts to split and divide because it thinks that it is going to grow into a new critter, and ultimately that's what happens, and the clone is born. Okay, so now there's a lot of videos on this. If you guys are curious, I would highly suggest looking at them because there's a whole bunch of stuff on this. I'll try to find some useful ones too. But it's a really clever way to not necessarily reinvent the wheel, but to make use of a system to get a critter that is identical to the donor. Okay. Now, this has actually been done for quite a while, and on smaller creatures. Every time I see this slide, I think to myself, I really need to update this. However, so humans have been doing this in the lab now, and they've cloned for quite a while, decades actually, and they've cloned tadpoles and carp and sheep and mice and dogs and cats and rats and horses and camels. And recently they actually managed to clone a black-footed ferret, which is an endangered species, and it's one of the first time an endangered species has been cloned. So when you're losing species left and right, this is potentially a great way to save them, okay? The problem is, though, that you're creating these new species or these new individuals, and they are identical to whoever donated the genes, okay? Now, other problems that seem to have happened is that Dolly the sheep didn't live as long as her predecessor. And one hypothesis for this is the fact that her DNA was old, technically. Okay, but researchers don't really know that, and this has actually been relatively controversial, and I've seen it argued in the literature, and I don't know what the final conclusion was. Cloning is definitely not the complete answer, but it is interesting and provides us with a relatively cool tool to potentially save endangered species and maybe even bring some back from the dead. Now, another way to potentially bring back species that have gone extinct or were domesticated is to actually use selective or artificial breeding. So if you think about it, we've talked about domestication, where we select for certain traits like tameness and friendliness and aggression, lack of aggression, that type of thing in our creatures. Well, if you go backwards and do the opposite, potentially you could, get combinations of genes that provide you with some sort of genetic look-alike, or, some sort of critter that looks like one that might have existed before. Now, it's not going to be perfect, okay, because this is a new combo of genes and you're artificially selecting. But back in the 40s and 1940s and so, this was actually used to bring back the auroch, okay, which I'll tell you an interesting story about them on the next slide. Now, what you do if you're trying to bring back an extinct species or something that looks like that, using selective breeding, is you potentially inbreed, okay, which is the pattern that goes to the left, or you can hybridize the pattern that goes to the right, and then you look for critters that are either similar or dissimilar, trying to figure out how you're going to get the ones you want, and then you can start crossing them to get these traits that they had, okay, getting the desired characteristics. Now, the reason we know this works was thanks to Adolf Hitler, believe it or not. And so, as you all know from history, he was an evil, evil man. Okay, and there's a reason I kind of chuckle at this, and I'll share that with you in a few minutes. But what he wanted to do is he was fascinated with breeding, and we know, unfortunately experimented on humans and did horrible things. But apparently he also had a breeding program for creatures that were extinct, and his goal was to actually bring back some extinct species. So one of the ones that his people worked on was the auroch. Okay, now they went back and bred and selectively bred and artificially bred and so forth and carried out their crosses, and they got a critter that looked a lot like the auroch. Okay, but this is the awesome part because, the Nazis were pretty evil, and I have a German background, so I actually get to say that. My family was German. But anyway, what was interesting about this, and kind of ironic, very ironic, is that these guys became so aggressive because they were, selecting for that they ultimately had to destroy part of the lineage because they started attacking all the humans. Okay, I'm sorry. That should not be funny, but they were attacking, remember, the Nazis. So, again, realizing that, history is what it is, and it's not always pretty. Part of me just kind of cackles at that because, they were not exactly the nicest batch of people. So Hitler and his people. But anyway, so, it's an interesting thing historically. Now, there are some of these that still survive, and if you guys want to look it up on Google, go ahead and take a look. And they're very controversial as far as whether or not people wish to keep them around or not. So they obviously kept the ones that were less aggressive, but some of them they did have to eliminate because they were just going after the people, trying to kill them. So, that's what you get when you start messing with the genetics. Now, if we do in fact start bringing back extinct species, there's a lot of potential candidates for this, and I'm going to do a little research and give you guys some articles and the announcements for this particular week. The passenger pigeon, the dodo, the woolly mammoth, and the quagga, which basically look like a cross between a zebra and a horse. Now, the reason these are potential candidates are humans are the ones that cause them to go extinct. And to be honest, I don't have a problem with bringing these guys back because it is hypothesized that we had something to do. But to do with their extinction. There are a lot of moral implications with this, okay? And on the next quiz, I'm totally asking you this question. Did I mention I'm going to ask you this question? Because you should be able to answer this next question on the next slide, because I'm totally asking you this question on the quiz. Hint, hint. Based on what we've talked about, what would be the moral implications of bringing back an extinct species? I'm going to ask you that, and you have to use information based on what we talked about in class to back up your answer. Now, there's no right or wrong answer, whatever your feelings are. Okay, if you think we should bring them back or not bring them back, I'm going to leave that up to you. But you do have to use information that we've learned from class to justify your answer. Okay, and as you're doing this, what I want you to think about is what caused them to go extinct. That should make a difference in your answer. Where would they live? Okay, well, there's limited resources. Shouldn't we focus on saving the species we have? Okay, so these are things I want you to think about because, as I mentioned, you're going to see this question again. Okay, now this lecture is a little bit shorter, and the reason is because there's a really good video, a cool video I'm going to have you guys watch, and it's from Hendrik Poinar. This is a lab at McMaster University in Canada. These guys are the ones who trained me in ancient DNA. Okay, so I'm a little biased. However, I know their level of science, and they're really amazing. And they also bring up some questions that I want you all to think about as you're addressing this here, okay? So the last part of lecture today, I want you guys to watch that video. If we were in class, I'd be having, physically in class together, I'd be having you do the exact same thing. Once again, if you guys have questions or you need something, please let me know. I'll be more than happy to meet with you. Stay safe, guys, and I'll talk to you again soon.